So as I was saying, mosses are difficult to identify. This is a picture I took that you see on the screen here of what we call a sphagnum moss. I guess I should write on the screen now that I never realized that the chat doesn't record. So when you watch a recording of that, this is sphagnum moss. This is Sphagnum. You see a lot of this around. Some of the stuff we looked at in class the other day was sphagnum moss. I find it a lot in the woods, especially after a good rainstorm. It's very plump, you know. I actually have another picture of some that had turned red. Apparently, it's not as common, but in sphagnum moss, if they're exposed to a lot of sun, they'll be more of a reddish color. But so that's well, not kind of. I was going to say that's kind of where we're going today. It is where we're going today. We're going to talk about plants, but we're going to start down here with a group of plants that don't make seeds. I don't know how many of you may have known that, but not all plants make seeds. We assume that plants all have, you know, flowers and seeds and fruits and all that kind of stuff, or nuts or something like that. But come to find out, there's quite a few plants out there that have neither flowers nor fruits, nor in this particular case, seeds. There's quite a wide diversity in plants, and that's why we spread this over 2 chapters. Actually, chapter 29 it'll be we'll talk about the seedless plants, and in chapter 30 we'll turn our attention and talk about the plants that do have seeds. Of course, that'll be something that we talk about next week. So let's go back and make the connection once again. So you came in the room here. Hi, Suzanne. How are you? Or she's coming in the room. Hello, Suzanne. $300,000 extant, that's the K, 300,000 extant species of plants. So this is a group of organisms. Hi. This is a group of organisms, eukaryotic organisms, that have done quite well for themselves at 300,000 species. The first, now this is where we get into this whole idea of the polyphyletic or the paraphyletic nature of protozoans. There's that chart again on the right. It's the same thing we looked at the other day. I've just highlighted one part, the part we're going to look at today. These are the land plants. The land plants are in a group, okay, of protozoans known as the archaeoblastids, which includes the red algae, which are considered protozoans, the chlorophytes, which are protozoan plant-like, charophytes, which are more plant-like, and then, of course, the land plants, where we're going today. Now, when all of this happened, these land plants, notice we're saying land. There are more advanced plants that live in the water, but they actually are plants that you know, evolved alongside of these land plants. Land plants we're moving out of the algae mats, we're moving into the, you know, the things eventually like we know today, the trees and the stuff like that. Now the chlorophytes, that's the Volvox, the Spirogyra, the green algae in general, the things that we think of it as the green algae that are more protist-like, they never made it to land. But a very closely related group of something called the charophytes, and there's a couple of charophytes down below. So charophytes still exist today. Remember, by now, I hope I've dispelled this idea that evolution works in this ladder kind of thing, right? That just because charophytes were the ancestor of land plants doesn't mean that the charophytes died off completely, and then there are land plants. The ancestor of the charophytes and the land plants, I mean, this critter right about there, yeah, that one probably has gone extinct, but there are still extant or existing charophytes, Cara nitella and Silicato or bicularis. They're on the right. Those are plants. Those are considered plant-like green algae or plant green algae. Remember, that line kind of gets blurred there. Now, where do we have evidences to suggest? How do we look at those kinds of things? You know, this kind of goes back to what we were saying at the beginning of the semester. How do we know how to build these relationships? You could almost think of putting together a cladogram with these things that I checked off here. These are some of the evidences that we have for why it was the charophytes that gave rise to land plants. And I'll let you look at those for a second. And I want you to be thinking about how is that an indicator of relatedness? So we're trying to build the case that charophytes share an ancestor with land plants. There's certainly an ancestor more recent than the other green algae, the chlorophytes. And we're looking at these lines of evidence. And the first one is a similarity in cellulose production proteins. Now, y'all know what cellulose is, of course, so that's not really the issue here. But if you think about what that's saying, a similarity of cellulose production proteins means a similarity in the primary level of protein structure. Now, if you remember back again from Bio 1, the primary level of protein structure is the amino acids, the order of the amino acids. And remember, proteins are all about shape. So if you don't have the right amino acids in the right place, holy cow, with sickle cell anemia, which has nothing to do with plants, but it's just one amino acid that's wrong, and you end up with sickle cell anemia. So if you don't have the right amino acids in the right place, you don't get the protein folding the right way potentially. And that means you affect the function of the protein. You get, put all those connections together. Well, now, if you remember at the very end of Bio 1, you kind of did full circle. You kind of went back and figured out, all right, how do ribosomes, how do they know how to make a protein the right way? Where's the recipe come from? Remember, that was the DNA. That was the DNA and the messenger RNA. And if you recall, there was a table. It was a code table. There was a codon table. And it said that you read the bases in the messenger RNA, 3 of them, and that coded for a particular amino acid. So backtrack through what this first little check mark says. If there's a similarity in the proteins that make cellulose in the charophytes, between the charophytes and the land plants, that means there's a similarity in the amino acid sequence of these proteins. And if there's a similarity in that, there's a similarity in the messenger RNA codon. And if there's a similarity in that, there's a similarity in the DNA. Right? Remember what I said back in chapter 25 when it comes to relationships? It's all down to the DNA. Remember we said that one thing they do is take DNA molecules and unzip them, right? Tear the letters apart and see if they'll stick back together. Because if the letters match, then you're related. I mean, everything stems out of that. There's nothing that you can tear down from further from that to suggest any difference. If your DNA is the same, then you are related, and of course those are relative degrees of things that are the same. Okay, there's some morphological stuff, like, for example, the sperm. Yeah, plants do have sperm, right? But there's the sperm similarities between charophytes and land plants. Both of them form this thing called a phragmoplast, which forms the cell plate. You learned about the cell plate in Bio 1. That's what divides the plant cells. After the cells have done their mitosis with the nucleus is divided, then the actual cell divides by building a cell plate, and there's similarities between those. And then I guess the last one is sort of redundant based on certainly what I said in the first one, right? Genetic analysis. Now, plants are something interesting to study. Obviously, people make their living off of plants, and people live off of plants. They're an extremely important organism in this environment, not just only for things we eat, but also for the things that we eat that eat plants, right? These are plants are what we call, or photosynthesizers in general are, but plants are certainly on land the predominant photosynthesizers. Very critical for an ecosystem, right? These are the organisms. You remember back again to photosynthesis. These are the critters that can take something inorganic like carbon dioxide with no little energy value and turn it into something organic, glucose, that we can burn for fuel. And they do that by using the sun. That's very important. We can't do that. They can. So talking about plants and being interested in plants and being concerned about plants is obviously important, because if you lose plants, you lose the ecosystem, and everything collapses from that point. All right, let's go back in time. Right, wavy screen like we're going back a little bit. Let's talk about this idea of moving out onto land. So I found this cool, really simple but neat graphic down below online, and I want to share it with you guys. So we're going from left to right in time. So on the left is the Precambrian. So this is before the Cambrian explosion, and then the Silurian, and then that's before the Permian, and then the Cretaceous is the dinosaurs. All right, so you get it, and then of course we're on the right-hand side. So you get an idea where we are. Very, very broad spectrum here. So in the Precambrian time, there were phytoplankton. Phytoplankton are photosynthetic plankton. Plankton, and we'll talk about this when we start talking about fish and that kind of stuff, but plankton means something floats. It really doesn't have anything to, well, it does. I mean, we don't really think about plankton as being these little things, and it can be that, but it can mean other things. But phytoplankton photosynthesize, so they're producers. You got the red algae that photosynthesize, you got the brown algae, but they're confined to the water. Remember, those are protozoans, those are protists. But look what is inching up That's why I like the way this is drawn. Look what is inching up, kind of out of the water on the shore. The green algae, right? The chlorophytes, which they stayed in the water, and then their relatives, okay, the charophytes found a way to get out onto land. They found a way. About 420 to 470 million years ago. All right, so right after the Permian extinction went on, these organisms, remember the Permian extinction, what we had was these continents forming together. So there was a lot of different habitats that were forming during that time, a lot of chances for different kinds of critters to find places to live, to eke out a living, right? Eke out a living. Now, one of the things that when we move out on the land that we've already talked about is this idea of desiccation, right? Drying out. That's a big concern. That's a major stumbling block for a lot of critters. That's a major stumbling block for critters being able to get out and get away from water and completely cut well, we're all dependent on water, but I mean, especially for reproduction, right? The amphibians still got to go back to the water to lay eggs. So when it comes to the plants, that's a little bit more difficult because plants can't move. And once you moved out on the land, if you're a plant, well, you're going to have to stay there. You can't pick up somehow and move on back to the water to lay eggs to raise youngins, to raise junior, right? So what these karyophytes that moved out on the land had to be able to do was make these very durable spore walls. Now this has to do with the reproduction part. Think about oh gosh, I don't want to go there. Think about a spore as a reproductive unit, a thing that allows organisms to reproduce. Okay, kind of. I didn't really want to go with the offspring thing, but you'll see what I mean as we kind of get into it. So spores and karyophytes develop these spores, very durable things. Look on the right, the little picture there. These are a couple of spores, spore tetrads. They come in groups of 4 or even individuals, depending on the species of plant. But these things are, I don't know how well you can see there, 470 million years old, 425 million, and these are still intact. Intact spores, right? So these are some pretty heavy-duty things, right? What is it about spores? Well, it's in the coating. Okay, just like when we looked at the the anthrax bacterium makes an endospore, and I said that was a durable outer coating. We talked about the brain-eating amoeba that makes a cyst, and I said, organisms, if you don't find yourself in the right environment or a harsh environment, if you can make some sort of a suspended animation thing, a cyst, a spore, then you can wait for the environment to change. Now, you can't wait forever, but you can wait a long time, okay? And the reason why karyophytes were successful in being able to get onto land and deal with these harsh environments was the fact that around their spores were this something called sporopollenin. Now, sporopollenin is one of those super duper heavy duty natural things out there that Mother Nature puts together. It's kind of like, did I mention lignin to you guys? Yeah, in the decomposers, lignin, the stuff that makes wood, allows trees to be very firm. That lignin is very hard to break down. There's only a few producers that can do it. Sporopollenin, you can see the little spores in that black and white picture. The sporopollen is what's covering the outside. Very durable to acids, bases. They're very durable to changes in pH, temperatures. Some of the research I've read about in looking into the spore construction and what it's made of, in order to remove the sporopollenin, they take the spores and they boil them in acid. Boil them, and then in acid. That's some heavy-duty stuff. So this is something very durable, right? This is something that can't live without water, okay? But if you make enough of them, they might spread, and maybe it'll rain someplace where the spores are, or the spores will eventually find their way someplace, through air or something like that. Anyway, through trial and error and over again, and differential reproductivity and all those things we've talked about, plants, these green algae begin to make their way out on the land, onto the land plants. Now, when they got on the land, of course, like we said, one of the problems was the desiccation. That was a real concern. But there were some good things too. So for every bad, when an organism moves into an environment, everything that it has to deal with or try to adapt with or go extinct, which usually happens, there are sometimes things that say, okay, well, this is actually a benefit of being here, because yes, there was the scarcity of water, there was the desiccation problem, but there's a lot more sunlight on land. Sun doesn't get filtered out through the water, so guess what? there's a lot more sunlight. If there is a lot more sunlight and you figure out a way to capture more of that sunlight, making leaves, right? If you can make leaves, and if you can make big leaves, you gain an advantage, because the more sunlight, the more photosynthesis. And in the atmosphere, there's more carbon dioxide too. Two things that you have in the water, yes, light to a level, of course, that you have in the water, but you have in greater concentrations on land. So you move onto the land, you're dealing with the desiccation, but holy cow, you get all this other stuff too. And there's a lot of decomposers on land too. So the soil is very rich in nutrients. So those kinds of things that you need, like your minerals, potassium, nitrogen, and all those kind of things that we've talked about, fertilizers, kind of, those sorts of things are in heavy concentration, very plentiful in the soil, all right? As I said, of course, the scarcity of water, but another thing that land plants had to deal with is this idea of being able to stand up, right? Now, being able to have some sort of a structural support on land, because that's not a big deal in water, but of course on land it is. So that's where plants started coming up with this idea of lignin, which they used to stand up the cellulose in the cell walls, all those things, that contribute to allow that plant cell by itself, that cellulose, to be firm and rigid. Very different than an animal cell, but a bunch of these together to be very firm so they can stand up. Because of course, all this unfiltered sun is nice, but you got to be able to get up to get to it. So if something's outgrowing you, then you're going to have to grow taller and faster, presumably, right? So that's kind of where we're moving left to right. Now, something that's interesting, and I throw this in now, and we'll talk about it more with other species later, but it always seems to me like organisms, they crawl out on the land, or these algae move out on the land and go, whoa, check it out, man, there's all kinds of resources. They start growing. They start getting bigger. They make bigger leaves, and they start becoming these big plants, and then maybe it gets crowded. Maybe this was a good place to start off, but man, I tell you what, now it's kind of crowded, and I'm feeling a little bit claustrophobic and moved in. So in some cases, we see organisms that actually move back into aquatic habitats. And what we mean by that, like, for example, mangroves and seagrasses. Mangroves and seagrasses are more closely related to land plants than they are to the organisms that gave rise to land plants. You see what I mean? So the fact that they live in the water, red mangroves like we have here in Florida, and turtle grass, eel grass, manatee grass, all that seagrass, these things are more closely related to, why does my pen do that, to the land plants than they are to the stuff on the other side of that hypothetical green hill there, right? So they return back to these aquatic habitats, and I think a lot of it many times has to do with the fact that reduced competition. Maybe now that you're an advanced land plant type descendant, you have some special characteristic that allows you to live in aquatic habitat very, very effectively. You're not competing with the algae anymore. You're different. You're a more advanced plant. Another organism that's done this, and we talked about it briefly in evolution, is the marine mammals, whales, dolphins, and porpoises. Remember, that's why they have those vestigial rear legs. They have in some species anyway, like the blue whale, you can actually find this vestigial part of a pelvic girdle, because these organisms used to live on land. So again, in the animals, and we'll talk about it, right? Same thing, out on the land, populated, everything went crazy, and then some went back into the water. Interesting. All right, let's talk about land plants in general, not just the seedless plants that this chapter is about, but just in general about the things that land plants did that if you could interview land plants and you could say, so land plants, what do you think were some of the keys to your success? Like, for example, what do you think were your top 5, and were there any others that you think were also contributed to your success? This is what land plants would say. They'd say this. They'd say, well, first, alternation of generations. All right, remember? And now I'm going to go ahead and go through this slide, but you are going to see it again. So if you're trying to follow along, don't worry the fact you haven't read the rest of it. I'm going to go through it sequentially. So alternation of generations, let me remind you what that is. Alternation of generations means that there are two multicellular stages. All right, There's a diploid and a haploid, like with us. There's a sporophyte and a gametophyte. But the fact what we mean in alternation of generations, if I guess we could call it alternation of multicellular generations. Another word for this lifestyle, by the way, is called the haplodiplonic. Did I spell that right? Haplodiplonic. Haplodiplonic also means alternation of generation. It means I'm trying to see if I can spell it right. Two multicellular, haplodiplontic, lontic, TIC at the very end, okay? Haplodiplontic. So haplodiplontic, alternation of generations, whatever word you use, they refer to the same thing. They refer to the fact, very different than in animals. That's kind of key that you got to know that, right? For one thing, animals don't make spores. So we don't have that stage right there. We do make zygotes. We grow up into something. We don't call it a sporophyte, though, a 2N. And through meiosis, we make gametes. So basically, we skip this part right here, right? We go from the sporophyte directly into making single-celled gametes. These organisms, through alternation of generation, the sporophyte undergoes meiosis and makes lots of spores. Now, over on the right, I put something in red I want you to remember, and I've said it a couple times. It's very important. When we talk meiosis, we'll talk about variability. Meiosis makes variable cells. That has to do with reproduction. Mitosis, we're talking about numbers. Mitosis makes identical copies of cells to replace cells. When you grow, right? That's what I mean about quantity. So when the sporophytes down in the lower left, when they undergo meiosis, think those spores are variable, very different, genetically different from one another, right? And those spores then undergo mitosis. So what we get is a bunch of different types of spores, okay? And then you take care of the fact that most of those spores ain't going to make it. Most of them are going to die. With those spores that are different undergo mitosis and make lots of themselves, all right? So we get lots of those spores. And then those spores, if they find themselves lucky enough, do germinate. They germinate into a single cell, then more than one cell, and then many cells, and they become a gametophyte. You might actually recognize this was the gametophyte we looked at for ferns. This is the prothallus. Remember that heart-shaped thing? All right, we'll see it again if you don't, right? Now, those multicellular gametophytes, as I said, are lots of antheridia, lots of archegonia. We got a factory going on there, so we're going to make lots of gametes. So we got lots of gametophyte tissue, okay, lots. We're saying that word a lot, lots. We got, on that gametophytic tissue, we got gametes being produced through mitosis. So we got lots of gametophyte, we got lots of antheridia, got lots of archegonia producing lots of gametes and lots and lots and lots, because guess what? In this case, it's a fern, but if you're like a moss or any of those kind of critters, most of those are not going to make it. Some of them will in fertilization, and we complete the cycle there, right? All right, multicellular generation. Both of them, two multicellular generations. Meiosis is variability, mitosis is quantity. Those are three real important things for you to remember about this, because as we go through different groups of organisms, especially through the plants, we see how how evolution has changed and modified this. All right, so let's take a look at some of these spores that I've been talking about and their durability. I showed you the other day in lab on the back of a fern frond in the upper left, those little dots. Those are sori, and those sori are sporangia. So that's where the spores are being made. So inside of those sori, and it looks like something on the right, actually, in the upper right, there's these little capsules, and the little purple things are the spores. So there's a bunch of these little sporangia. There's a bunch of these little sporangia in each one of those brown dots. Those sporangia each make a bunch of spores. Here's some spores. And in the case of the fern, for example, the wind blows those spores, and there's lots of spores going all over the place, hopefully to find a place to survive, away from the parent. Because that's one thing about offspring dispersing. You want them to disperse so they're not competing with the parent for resources. Get away, go. The one on the lower right, Equisetum, that was the horsetail. Remember the horsetail plant we saw in lab yesterday? Equisetum spores have little springs on them, elaters. Boom, and it shoots them out, even disperses them further. So it's all about the dispersal. Okay, so alternation and generation and spores and all that sort of stuff all fit together with this idea of organisms that are, going to be well adapted. Organisms are going to be very adept at moving into harsh environments. Okay, harsher environments. You don't find mosses and ferns living in extremely harsh environments, but certainly moving out underwater. That's what I mean by a harsher environment. Okay. All right, and we're doing that by, of course, like I said, increasing the numbers, and of course, variability is important because you don't know which genetic variability is going to work. All right, so now the second trait is something called multicellular gametangia. All right, what are gametangia? Gametangia is another word for the gamete part producing of a gametophyte. All right, you'd be like, whoa, too many words at once. Let me go back for a second. The green thing that you were looking at under the microscope for the fern, that little heart-shaped thing, is the gametophyte. It's a prothallus. It means it's kind of leaf-like, but that's the gametophyte. The little lumps and the bases and the antheridia and the archegonia that were on them, those are the gamete-producing parts. Those are the gametangia. So what we see in bland plants is the idea of multicellular gametangia. So gametangia that are big and can produce lots of gametes. So we didn't look at Marchantia under the microscope, but you probably will recognize, some of the way the picture is done here. There's the antheridia. In our picture of the moss, the antheridia, or in a microscope, the sperm were purplish or gray color. I do have a picture of it later on to remind you. But this is the male part. In the liverwort, the gametangia are a little bit bigger. You don't have to use a microscope to see them. The male ones come up and sort of like our flat surface, and then the female ones almost look like a miniature palm tree. See that? Now the archegonia on Marchantia are on the bottom as opposed to the moss. They were facing upright, but they still have an egg in them. So the sperm are going to make their way from the antheridia to the archegonia. Right, and with this idea of having multicellular gametangia is we're producing structures that are big enough now to make again, it's all about numbers and variability, of course, right? And variability. We got all these different kinds of spores that are producing these different types of gametangia. But really, when it comes right down to it, it's about hedging your bets, isn't it? Right? It's about producing lots of offspring, lots of gametes, lots of offspring. Okay, now here's something interesting. It's an addition to, or it's this way to kind of help hedging your bets. So far, what we've kind of focused on is critters, organisms making lots of offspring, lots of different offspring. But we haven't really discussed too much, I don't think in any of them that I can remember up to now, we haven't discussed this idea, what if you were to take better care of your offspring? So instead of making so many of them, why don't you make fewer, which would cost you less in resources? But why don't we do something like take better care of the embryos, take better care of the offspring? In other words, raise the offspring a little bit. And if you're kind of thinking, well, yeah, that's kind of what we see in mammals and humans. Yeah, right. We don't have lots of offspring. We typically have a few. I don't know what the average number is. But of course, for one thing, the offspring develop inside the female. So that's protective and nurtured in our particular case. And then once the offspring is born, depending on the critter, there are many years that offspring is dependent on the critter. So what we see among animals, and we also are going to see among plants, is this idea of a dependent embryo, a multicellular, so a larger but a dependent embryo, an embryo that is dependent and protected by the parent, all right? So in this particular case, just like it is in most cases, it's going to be the female. So the embryo is retained inside of the female gametophyte, right? And inside of that female gametophyte, in some cases, we actually get something really kind of cool, because it's like animals. We get this idea of placental transfer cells that actually nourish the embryo. You can see them in the lower right, the picture, the blue. Those are these cells are wound in the maternal tissue, the female gametophyte, and the embryonic tissue are wound together. So there's a lot of transfer of nutrients back and forth. There's a lot of diffusion. So you can see the embryo and then the maternal tissue. These are called embryophytes, these kind of plants that do this. Now, not all plants do this, but we are going to see as we go through talking about the plants, more and more emphasis on doing this. More and more emphasis on protecting that embryo. It's kind of neat, though. Like I said, in animals, we see something similar, too. When we get to the animals start talking about, we're going to go through a similar kind of progression of producing lots of offspring to making a few and taking care of them. That's why I write, who else does this, right? Animals do it. It's a natural response, if you think about it, with evolution. Survival of the fittest, producing the right variability. You could do that in a balanced way by producing lots of offspring with little to no parental care, all the way to very few offspring with lots of parental care. And when we look at organisms out there, that's exactly what we see. We see a gradient among animals, among plants, among organisms that take care of their young, or provide their young, like I always call like a starter pack, like develop inside the female and such. Fewer offspring when you do that, and organisms that don't provide any starter pack, pretty much all they provide is sperm and egg, blah, blah, blah. You're on your own after that. They produce lots of offspring, right? Makes sense. Makes sense. Spores, walled spores inside of the sporangium, right? We talked about the spores again, but now we talk about the fact that inside of the sporophyte, this is a cool one. This is the sphagnum moss sporophyte. So the picture I showed you at the beginning, oh, there's a red one right there, I forgot. The picture I showed you at the beginning was the green parts. That was the gametophytes. Remember, in mosses, it's going to be the sporophyte that is going to be small and dependent. And on this one, look at that. The sporangium is real obvious. Remember the ones we saw in lab yesterday? There were those wispy little things sticking up. And right at the top was the sporangium in those cases. And then there's a great little picture there, the microscope picture. Look at all the spores inside there. See? Numbers, 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 numbers. So we have these multicellular gametangia. We have these massive spore-producing structures. So we're really going to make sure that we throw everything out there in the hopes that something stays alive. It kind of reminds me of, organic chemistry. Throwing answers out there just to see if something works. All right. Another thing you got to worry about when you move on to land, knowing up and down. It's not a real big issue in water. I mean, organisms do have ways of sensing the bottom of the ocean and the top of the ocean, hopefully, and I'm sure, and they do. I was going to say I'm sure they do. I know they do. But plants have to have that ability to do that, much more so out on land than in water. So the fifth key trait for land plants, derived trait. So this would be an example of a synapomorph, by the way, right? Synapomorph, a derived trait, is the fact of these things called apical meristems. All right, now, apical, we talked about an apical complex in the alveolates the other day. That was at the tip. I said apex or apical is the tip. And plants have two of those tips. They have one at the top, which is called the shoot on the right, and they have the one at the bottom, which is called the root. Now, you may not remember, but if that picture on the left of the apical meristem of the root looks a little familiar to you, if you look a little bit closer at it, you'll say, I saw that. Yeah, in Bio 1, when you were looking at mitosis, you were looking at slides of onions, the root tip, the apical meristem of an onion. The reason why we use that to show you mitosis is that's where most of the cell division is going on. Most of the cell division is going on in the area of organisms where they're growing, right? It's one of the purposes of mitosis. So that's why those make great slides. But what is going on in terms of the plant? They're not sitting there making these great mitosis slides for us. They're growing. They're growing down and they're growing up. And they have to know that. They have to know that. They have to chemically, physiologically be able to understand that to be able to go in the right direction. Because you want to go down with the roots to anchor, absorb nutrients, absorb water. You want to go up, right? Because you want to be able to have sunlight. And disperse your young, because the taller you get, yeah, the onion cells, right? The taller you get, the more you can disperse your young. I guess that's what they would probably say. I think plants would probably say those are the top 5 characteristics for land plants. But there were a few others, right? I had listed 3 others. Something called a cuticle, stomata, which you probably may remember from Bio 1. We talked about in stomata during the dark reactions, specifically what C4 plants do with their stomata and C3 plants. So what's the cuticle first, though? The cuticle is the outer covering that you find sometimes on plant leaves. And I put a picture up at the top. Those are red mangroves. very common Florida plant along the ocean. Red mangroves grow with those huge prop roots. I always tell my students, if you're ever snorkeling around there and you want to see some really cool stuff, like a lot of nursery, a lot of small fish, go snorkeling up into those red mangroves. If you snorkel up into those red mangroves, that's the kind I was talking. Oh, hi, Olivia. I'm sorry. That's what I was talking the other day about those kelp beds. There's lots of critters that hang out in there. Anyway, a lot of times red mangroves very commonly grow in what we call brackish water. Brackish water is neither salty nor fresh. It's somewhere in between. It's not salt water, it's not fresh water, but it's enough salt that it does cause dehydration problems. So one of the things that the red mangroves do is they cover their leaves in that cuticle. In the lower left is a cross-section of a mangrove leaf. And the CU, is that CU? Yeah, that's labeled. That's the cuticle. Think of a cuticle as being like chapstick, the wax you put over your lips to prevent them from drying out. That's what the cuticle does. Plants can lose water through their leaves. It's kind of like, I say it's kind of like sweating, but it's not exactly the same. But they lose through their leaves, and so if you have a cuticle covering, then you can protect some of that water loss. So that would have been beneficial to plants moving on land. Down in the lower right, those are some pictures of some stomata. These were the openings on the underside of a leaf of a plant, and they allowed carbon dioxide in. Remember, carbon dioxide is necessary for plants, obviously for photosynthesis. Now, remember what I said a few slides ago. When plants moved out onto land, one of the benefits they got, bad thing was the drying out, but one of the benefits they got was more carbon dioxide. So guess what? If you can make more stomata, bring in more carbon dioxide, you're going to have an advantage. See, it all goes back to evolution, doesn't it? It all goes back to survival of the fittest and who develops these unique innovations to have just a little edge over your partner. What was the last one? What, mycorrhizae? Yeah, mycorrhizae. Mycorrhizae is a fungus. In the upper right in the brown picture, the larger items are the root of a plant, and the smaller little threads going in between are the parts of a fungus. We'll talk about what those parts are called when we get to fungus. But fungus, as I mentioned in class, I think already, fungus are unique in the sense that they digest their food on the outside. So not like us, right? They don't bring it in their stomach and then mix it with enzymes and digest it. They basically secrete enzymes into the environment, digest the nutrients, and then kind of suck them up. Well, they obviously secrete more enzymes, or they digest more food than they can take up themselves. And so anything growing in there has the advantage of having this pre-digested food. And one of the things growing there, of course, would be the tips or the roots of plants. So mycorrhizae in plants actually have a symbiotic relationship. Yeah, they actually live together. And it's so important that among vascular plants, and not all plants are vascular plants, but they're predominantly ones you think about, like flowering plants, pine trees, those kinds of things. 90% of vascular plants have mycorrhizae. They have these fungus that's living around the roots in that symbiotic relationship. The mycorrhizae gains something from the roots of the plant. And of course, the plant gains the benefit of having these mycorrhizae. This would have been beneficial to plants moving out on the land too. A harsh environment, right? An environment which you get a scrape out of living, you could gain an advantage, just a little advantage, if you came out on the land, you and your fungi buddy there, because the fungus is good at ABC in its food, right? Already been chewed. It's really good at breaking down nutrients in the environment into simpler forms, so myself, the plant, can grow, right? All kind of works together, right? It's amazing to me how sometimes when we look at the way organisms and how the evolution of life, and the fact that it happens randomly, I think always just blows me away. But the fact that these things kind of come together just at the right time, the fungus, the plant roots, and moving out on the land and all that sort of stuff, all right? Neat stuff. All right. We got any questions for me? Because if we don't, I think we're probably ready to move into the different kinds of plants. So as you can see, what we do in these chapters is basically we talk about some of the general characteristics of these, like the prokaryotes and the protozoans, and then we talk about different kinds of them. So let's talk about the plants. If you look at your attention up to the top, so we're talking about land plants. First, we're going to talk about the non-vascular plants, something we call the bryophytes. Now, that word is going to be a little bit confusing because it has two definitions to it, but I'll get to that in just a second, okay? So the bryophytes are non-vascular plants, and that's the three we're going to talk first, the liverworts, the mosses, and some called hornworts. And then we'll move into vascular plants, and we'll talk about the seedless ones first. So we got seedless non-vascular. We have seedless vascular. All right, so the non-vascular ones have no seeds either. And then finally, at the very end, not in this chapter, but in subsequent chapter, we'll get into the seed plants. We'll get into the gymnosperms, which you can see the picture of those, the pine trees, and then everybody else. Now, the angiosperms, that's the biggest group. Look down below. There's 250,000 species. So once plants got to that point in evolution, man, it helps that this was the dinosaur extinction. The dinosaur extinction helped a lot of flowering plants to diversify. That's why we get all of that. All right, so we're going back about 470 million years ago to the origin of the land plants, and we're going to move off now to the bryophytes. So the bryophytes, when we think about bryophyte as a collective term, we're talking to the seedless, non-vascular plants. Seedless, non-vascular. These are what we call the non-tracheophytes. A trachid is a transport cell. A trachid is a cell that forms a tube. You have a trachea in your neck, right? A trachid is, so those transport cells, the vascularization I'm talking about, you have your blood vessel, your cardiovascular system, right? Well, in plants, they have a vascular system. It's not for pumping blood, but it's for moving water, moving nutrients around. But the bryophytes don't have a vascular system. They're non-tracheophytes, non-trachids. So that means something. And it explains why we find them where we do. And it explains why there aren't any big, huge mosses like you see in the picture behind there growing somewhere. Because if you get too big, right, diffusion and osmosis becomes a problem. Diffusion and osmosis can work for a thin layer of cells, but it can't work for a thicker layer of cells. Because the cells on the inside, diffusion and osmosis won't be able to penetrate deep enough into the organism to provide those cells with the diffusion and the osmosis that it needs for reasons that I'm sure you remember from Bio 1, right? So those cells need to be able to do diffusion osmosis. Now, if you get really big, which organisms obviously do, what you do is you have little vascularizations that go between all those cells, little transport ways to make sure that all those cells get everything they need and, get rid of the waste that they need. Bryophytes don't have that. Bryophytes don't have those little tubes. So what do? Well, they're limited. They're limited to diffusion osmosis. So that means they need to stay small. So that explains why when you walk into the woods, you don't see big bushes or big trees of mosses. Mosses are usually low, to the ground, small, right? It's all about physics. Remember, living organisms, life is dependent upon all the laws of physics and chemistry. Those laws were in place, the ones in physics, then the ones in whatever, long before living organisms came on the scene. We have to obey the laws of physics. Diffusion osmosis is a law of physics. So organisms respond. Obviously, they do very well, right? Because there are mosses around. So again, it's not like a bad thing or a good thing. As long as you can find a place to live, darker environments, cooler environments, areas we don't have to worry about drying out, areas where it tends to be moist, where you find mosses, right? So this is kind of what we think maybe the first land plants looked like, mosses moving out on the land. Okay, of course, they have a gametophyte and a sporophyte. We looked at that in lab yesterday. The gametophyte is going to be larger. It's going to be more conspicuous. I think conspicuous is maybe a better word than dominant, even though I still see that word dominant used a lot. You know what I mean? Conspicuous. We looked at the moss. You look at that picture. You're looking at gametophyte. Sporophytes, again, were those little tiny wispy things, right? Those little brown things that we saw sticking above a couple of the samples of moss that I had. They're going to be smaller, very short-lived. Their job is really only to make the spores. And not only are they very short-lived, and not only are they not prominent, they're actually dependent upon the gametophyte nutritionally. So remember, we looked at the vase, and there was an egg at the bottom of the vase in some of them. And I said the sperm swims down there, fertilizes the egg, and out, this is the moss now, out of there grows the sporophyte. but it stays connected to the gametophyte and is always, it's not the same as an embryo here. Don't get this confused with embryophytes, right? That's the offspring. This is the sporophyte, the thing that's going to make the spores. This isn't the offspring of the organism. A little different with these guys. The spores the sperm are flagellated, so these critters are going to live where there's water because that's how the sperm get from one to the other. They swim through the water. All right, so now let's look at the 3 different types of bryophytes. The first ones are called the liverworts. I think that's a cool phylum name, especially if you remember that hepatic is liver. The Hepatophyta. Remember what I told you in class? These liverworts with their thalloids or their leaf-like structure, to some folks look like a liver, so that's where the name came from. The gametangia, the multicellular gametangia, we showed you before, those are the little things that look like palm trees if you're female, they're more like a tabletop if you're male. The sporophytes are actually contained on the same structure, the multicellular gametangia, as the female gametophyte. You see the picture on the right? So this is kind of a protection of spores, because here's the female gametophyte, right? But the female gametophyte also has the sporophyte hanging off of it. So the sporophyte, where the spores are made, is contained right in that female gametophyte. All right? So those are the liverworts. Hornworts. Hornworts get their name because of the fact that their sporophyte looks like horns sticking up. All right, what does it mean? No seta or seta. That is a. Let me change my color pen. When something has a stalk, if you will, and then say something at the top, we call this the setae or the seta. And this, if we're talking about a sporophyte, would be the sporangia. All right, now, the reason why it says in hornworts there is no seta is because all of that is sporophyte. All of that is sporangium. It's not a stalk that rises up to something at the top. All of that entire structure there, that green structure, because in this particular case, the sporophytes are photosynthetic. Remember the ones in the mosses, when they turn brown, they're not photosynthetic, but these guys, the sporophytes, are photosynthetic. Not only are they photosynthetic, they also have stomata. So these guys can help out with photosynthesis. All right, but that's what we mean by the fact that it has no seta. It has no stalk holding up. It's all sporangia. All right, there's the gametophyte down on the lower right. Okay, hornworts are a member of groups of organisms we call pioneer species. So, and I'm going to use that term maybe a few times for the rest of the semester. Let's talk about that. Pioneer species are organisms that can move into new habitats first, areas of new land, like when volcanic eruptions occur and enough occurs that stuff breaks through the surface of the water, or if there's a landslide that comes through and wipes the side of a mountain all the way down to the rock, or any of that sort of stuff. As the land begins to recover, as the land begins to populate, like in the case of the island, there are some critters, usually hardier critters, that can deal with these kinds of harsher, austere environments that do better than others, and we call them pioneer species. And there are pioneer species of plants and animals. So hornworts would be an example of some of the first plants you would find in these areas. And not only are pioneer species some of the first, but they're also good for paving the way for later species. We talked about this with the pollinators and the insects, how it's kind of this, working together. So hornworts have a lot of nitrogen-fixing bacteria, a lot of those symbiotic nitrogen-fixing bacteria, which we talked about, right? Which is good for plants because plants need nitrogen. It's limiting to their growth. So these guys move out into these sort of nutritionally deficient environments, and they get a foothold because they can do that, and then they actually start producing, or the bacteria start producing a lot more nitrogen in the soil, which means other plants can now get a little bit of a help, if you will, from that amount of nutrients. What the cool thing about that, of course, is you never think that the hornwort is doing it for later plants. This whole nitrogen-fixing bacteria and other plants taking advantage of it all just kind of arose through evolution. Oh, by the way, in your field notebook, you do have to have a bryophyte, and if you see one of these, I see them sometimes growing in the I see them in here in Florida, like in some of the ditches along the walkways and stuff. And their common name, they're called hornworts. But don't be fooled; these are not really hornworts. So the plants that look like this. So don't put that in your field notebook as a hornwort. It's kind of the same I said the other day with the mosses, or with the Spanish moss, right? Okay, so now let's talk about the mosses. We've gone through the liverworts, the hornworts, and the mosses. The liverworts, by the way, the phylum name, Anthoceratophyta. Antho refers to like antlers, so that's the horns that stick up on that. All right, bryophytes. Let me go back to this slide for a second because I want to show you. Bryophytes refers to two things. It refers to something in general. It refers to non-vascular plants. We've gone through the liverworts and the hornworts, but bryophyte, or the phylum Bryophyta. So bryophytes are liverworts, hornworts, but in the phylum Bryophyta, it's just the mosses. All right, so when you say Bryophyta, you can be referring to two different things. You can be referring to mosses and liverworts and hornworts, or you can be talking about the phylum Bryophyta and just be talking about the mosses. Okay, so sometimes these terms get confusing. So here's your typical moss. This is sphagnum again, and you can see the gametophytes, and you can see the sporophytes. You see what I'm talking about in that sporophyte? The capsule at the top, and then the seta is the stick that's holding it up. Different than we saw in the hornworts. Okay, very small gametophyte. There's the gametophyte on the right-hand side. Look at the scale. This is what, 0.2 centimeters. So we're talking what, maybe 0.2 millimeters. So we're talking somewhere about maybe 1.5 millimeters tall. Really small. Small little leaf-like structures around a stem-like axis. At the top of those leaf-like structures, folks, just to remind you where we were yesterday, at the top of these little structures are the Antheridia and the Archegonia, depending if you're male or female, gametangia. You see, these little green things that are growing up, they're gametophyte. The whole thing is gametophyte, but the little individual ones are called gametangia. The same thing like we saw in the umbrella and the flat structure in the hornworts. What do we have in the mosses are these little green wispy things, and they contain male or female gametangia. And in the male-female gametangia have the Antheridia and the Archegonia. All right? Sporophytes, again, brown. They don't photosynthesize. They are photosynthetic for a while till they start producing spores. Their primary job is to produce spores. They photosynthesize because while they're waiting to produce spores, if you ain't busy, do something. So these sporophytes are growing. Let's let them photosynthesize and help with that until they start making spores. Got to live in water, around water, though. Your sperm got to swim. They swim through little droplets from one Antheridia off of one male gametangia to another. And I think I mentioned the other day, one of the ways that they prevent, if you think about it, you don't want the male gametangia. You want to prevent inbreeding. So what happens is sometimes in organisms, the sperm will be ready and actually be dispersed, and then the eggs become ready to be fertilized a little bit later, giving a chance for the sperm to move away before the eggs are ready to be fertilized. These are not mosses. Spanish moss in the upper left, one in the lower right I've showed you in class is a lichen. It's sometimes called a deer or a reindeer moss. But again, that's not a moss, don't forget. All right, let's take a look at, this should be, at least the pictures should be familiar from yesterday. The pictures in the upper right, the microscope pictures that we see right there. Right. So on the left-hand side is the Antheridia, the darker purple, right in here. It's going to be the sperm. On the right-hand side is the Archegonia. I got a little vase and an egg is inside there. So let's kind of put that together with how this all works in terms of alternation of generations. So where do I want to start? Let's start with the male and female gametophyte. So let's start right here. So we have a male gametophyte, we have a female gametophyte. The male gametophyte contains the Antheridia, the female gametophyte contains the Archegonia. Now they may be on, the Antheridia and Archegonia get together might occur on different moss individuals, or you want to prevent it, but sometimes it happens on the same one. But scattered throughout that moss is Antheridia and Archegonia. Okay, Antheridia produce sperm which have to swim through water. They got to have water. A little drop. They swim from droplet to droplet until they find an Archegonium, And it's all chemical cues that bring them together. They swim down the archegonium and they fertilize the egg inside of the archegonium. And there's the zygote. There, if you want to think about it that way, because it's not really exactly the same, there's the offspring of these organisms. There's the result of fertilization. There's a different critter that's being made. There's some variability. And the zygote grows as an embryo, and then that sporophyte, which remains dependent on the gametophyte nutrition, grows out the top. So that's what we were looking at in lab yesterday with those little sporophytes sticking up. Those sporophytes were sticking up among these little short gametophytes and the little brown wispy things with the capsules and the setae and the sporangium. Then this is where the spores are going to be made. Now, meiosis is going to produce the spores. Meiosis, then mitosis. Meiosis for variability, right? Variability, then mitosis for lots of them, up to millions, 50 million of them in some cases, and we're going to disperse them. Now, dispersal is important. You might have a little spring that shoots it out like we saw in the horsetail. Look at the picture in the lower left. This is the peristome. The peristome is the opening of the capsule, the spore capsule, where the spores are going to shoot out. See the little finger things? They look like, how a diamond is held into a ring, whatever, prongs. That's used for spraying the spores. It'd be like if you held your finger over the end of a garden hose. Isn't that cool? It's like if you put your finger up and the water would spray out. That's what that's for. So you get the maximum dispersal of these spores, man. Lots of them, too. Millions of them. Then there are, in this particular case, in these species, there's a male spore and a female spore. Right? So we have a spore that's going to produce the male gametophyte, which is going to make the antheridia, and then a female going to make the female gametophyte and the archegonia. Bud. What's a bud? A bud is just a germinated spore. First, it starts off as a protonemata, which is just a few cells. See, that's already multicellular. And then off of that protonemata comes a bud, which has an apical meristem, so it knows how to grow up. Important, right, for plants? All right, so that's how mosses do it. So that's it for the non-vascular plants. Let's move on now, if there aren't any questions, to vascular plants. So now we got these organisms living on land, but sort of limited, right? They're not very big. Diffusion and osmosis, is really all they can use in terms of moving nutrients. They don't have vascularization, so they're kind of limited to some pretty select environments. Cooler, moisture environments, and they're going to stay small. They're not going to get very big. They're not going to grow large. All right, but that's going to change now because plants next came up with this idea of vascularization. Or maybe not next, I shouldn't say, but a lineage of plants came up with this idea of vascularization. So the mosses and their kind were around for quite a while, 100 million years, pretty much with the place of themselves. About 425 million years ago, we start to see in the fossil record vascular plants, like the one on the right, Cooksonia. That's a fossil of Cooksonia, a drawing of Cooksonia with its sporangia, and then the lower right is a computer-animated picture of what we think Cooksonia may have looked like. On the left, then, is an artist's recreation of the Cooksonia. Or not Cooksonia, I'm sorry, but another early vascular land plant. This was Aglophyton. Interesting thing about this, for one thing, is we have this very dominant now, or very non-dependent sporophyte, an independent sporophyte, getting bigger and larger. Now, these plants weren't very big. For one thing, in terms of they couldn't grow very big, even though they had vascularization, because they couldn't make enough food. The only photosynthesis they could do was the parts that you see there in the green. The sporangia part was the part, obviously the part that produces the spores. Now, they didn't have stomata, so they could take in carbon dioxide, but they were still kind of limited in how big they could get. They were only a few centimeters tall. They did have rhizoids, and let me make it very clear now, there can be confusion. A rhizoid is not a root. Not a root. A root does two things, okay? A root it anchors the plant into the ground, okay? And a root also has vascularization. A root has an ability to suck things up from the ground. We know that's where they get their water. A rhizoid, so in these early vascular plants, a rhizoid only does anchoring. So organisms, plants all have rhizoids, which just anchor the plant. They don't suck anything up. Now, the bigger plants, more modern plants, if you will, or derived plants, they have roots, and roots do both. They do the anchoring of the rhizoids, and they do the absorbing of the nutrients as well. Now, don't confuse a rhizoid with a rhizome. A rhizome is a root that runs horizontal. That's a rhizome. All right, now, keeping on with this idea of how these organisms have adapted to their environment, what I want to look at next are what are some of the things that vascular land plants. So, as we move from some of the basic innovations of the bryophytes, the multicellular gametangia, the dependent embryos, the knowing up and down, knowing the apical meristems, what did the vascular land plants do? What did they build on, if you think about from that point forward? And again, I hesitate to use those terms because it kind of implies there's some sort of a hierarchy to the, who's better than who is in a plant, and it's not that way. But we're saying among certain lineages, what were some of the advances that they may have come with to allow these organisms to survive in different habitats? And so I got 5 of them listed here. So this would be the same thing. If you went up to vascular land plants, you'd say, what are your top 5 characteristics? Now, I just noticed that I don't repeat these slides, this particular slide, but that's okay. I'll refer back to it. All right, number 1, the sporophyte is becoming dominant. We already see that. We already start to see that in some of the plants, but we see it much more readily here, especially when we look at some of those fossilized plants I was just talking about. But the sporophyte becomes more dominant, becomes more conspicuous, it becomes independent. So I got these 2 pictures side by side to show you what I mean. There's the moss on the left. The gametophyte is the green, more conspicuous. The sporophyte's the small little wispy things. And then in the fern on the right, the gametophyte is this thing. This is that prothallus you were looking at. This is that little tiny thing you were looking at underneath the microscope. And what this little thing growing out of it's going to be, that's going to give rise to a fern frond. And this is the sporophyte. Because on the back of many of them, they have sori, which produce spores. Sporophytes make spores, gametophytes make gametes. All right, so on here there are Antheridia and Archegonia, and in the Antheridia of one fertilized the Archegonia of another, and out came Junior. That's Junior. But you see what we have now in the tracheids, the vascular ones, we have much bigger sporophytes. Because you've got vascularization, you're not limited to growing small. You can grow tall. And when you can grow tall, you can get up above everybody else and photosynthesize, and you can disperse your young. It's kind of like the evolution of the eye. Every little thing along the way has got to provide a benefit for the organism. All right, since we're talking all about vascularization, let's take a look at it. Now, the vascular tissue that I'm talking about, or the vascular cells in a plant, are called the xylem and the phloem. And the xylem are tracheid cells, and they move material up. They're lignified, and the xylem is going to be usually on the more toward the center of the plant or the tree. So there's the xylem here. And the phloem, which transports things like sugars and amino acids and produced material throughout the plant, is usually found up near the edges. So this is phloem here. So I always remembered xylem up, phloem throughout. Xylem brings the water and minerals up from the ground. So what we're looking at here obviously is a cross section. We're looking at if you took the stem of a leaf or a plant and you cut it real thin and you looked at it that way. So now you got the ability to grow tall, right? You're a tracheid. You can outcompete. You can outdisperse other species. Again, it may not have been an earth-shattering change, but it was just enough to give you an advantage, right? So we have the sporophytes becoming dominant, and one of the reasons they're becoming much more dominant is the fact that we got tracheids now. We got these vascular tissues, so these plants can grow tall. Now we're also going to have true roots too. So roots that are going to do anchoring, just like rhizoids did. There's the word rhizome, so you don't get confused. Rhizome is a horizontal root. So this is a true root, like we're defining them here. My pen, goodness gracious. It's supposed to be root, R-O-T, whereas a rhizoid is just something that anchors. There's no absorption there. So you see down below in that fern what you got there, these little rhizoids sticking out. So anchoring and nutrient and water absorption occurs in roots. Now, rhizomes, may have been something that gave rise to rhizoids. So maybe we had roots and then we had rhizoids, or really when it comes right down to it, if you think about it, considering these ferns and organisms possessing rhizoids would have come out first, that's a good evidence for convergent evolution, even down at the plant level. All right, now the leaves. As I said, you move out on the land, you can take advantage of more sunlight by making larger leaves. So you increase your surface area for photosynthesis, and that's obvious what that's going to do. So, and of course, that makes more stomata, which more carbon dioxide. So let's look how that happens among plants. There are two basic ways it occurs. Among species of plants, there are those plants that have thin, unbranched leaves, like you see on the right, the microphyls. That Krause's spike moss, which is an organism still around today, is a lycophyte. And lycophytes have small little leaves with unbranched vascular tissue, microphyls. You can see how microphyll comes about in the bottom there on the left. You got a stem with vascular tissue. So mutation or whatever occurs, we start to get this offshoot. And now we have a little leaf that expands out, and we got a little increased photosynthesis. That's pretty good. But let's go one step. What if we taken plants, like you see down below, see that branching stems with the vascular tissue? I said that's what whisk ferns look like. So what we have is that unequal branching. So that means the branches aren't in the same plane. And then we have a mutation that brings all the branches in line with each other, and then simply fill in, kind of like the bat does between its fingers with flight membrane, so a wing. We actually fill in between all the branches, and now we get bigger leaves. And those are called megaphylls, like we see in that fern you see on the right-hand side there. Now, so bigger leaves are good, right? Yeah, they're going to allow better photosynthesis or more photosynthesis, take advantage of more sunlight. But now there's going to be kind of a balance that goes on. About 360 million years ago, the atmospheric carbon dioxide levels began to drop drastically. The Earth's atmospheric carbon dioxide levels began to drop. If you could make bigger leaves, so think about how this works sometimes in evolution. More carbon dioxide make bigger leaves to take advantage of. Carbon dioxide levels drop. In some cases, what plants can do is make bigger leaves because they have more stomata to absorb that carbon dioxide. So it kind of depends on what these plants do at that time, because remember how these mutations occur, right? It's sort of a random. But now when you get larger leaves, if that's your answer, you got another set of problems, and that's overheating. And plants can't overheat because their enzymatic reactions, like ours and everything, have a very optimum temperature. So what you have is now more stomata, larger leaves absorbing more heat, but the more stomata not only allows to take in more carbon dioxide, but the more stomata also allows evapotranspiration to occur. Evapotranspiration is what I said about with the red mangrove, them losing water off the leaves. It's kind of like plant sweating because it prevents overheating. Now, if you're looking at that thinking, oh, it seems odd because it's actually not good to be losing water, it isn't. It's a balance thing, right? We sweat, we lose water. We don't want to do that, or what we do to cool off, but that is precious water. That's why if you don't rehydrate when you sweat a lot, you're going to run into problems. So, overheating is an issue which you try to mitigate by this evapotranspiration problem. But of course, that's a balance kind of thing. And larger leaves would have brought in more stomata, but also more heat, right? Because there's more for the sun to shine on. All right, sporophylls and spore variation. A sporophyll literally means a spore leaf. The parts of the plant that make these spores, and eventually we're going to start talking about the parts of the plant that make the sperm and the egg and all that sort of stuff. We move on to other plants. Evolutionarily speaking, those are modified shoots, modified leaves. Those are modifications of plants. Things like pine cones, like you see in the picture on the right, the sori, and all the flowers, when we get into those are just all modifications of leaves. In the case of the sporophylls, the spore leaves, these leaves were modified to contain the spores. In the ferns, it's the sori, or the sorus if it's singular. In lycophytes, we just talked about them with their leaves, and in gymnosperms, cone plants, pine cone trees. Pine cone trees or pine trees are gymnosperms. They don't produce sori, they produce a strobilus or strobili. Strobilus is from Latin cone, they produce cones. So down on the right is a lycophyte with strobili. That's the cones they make. And then this is a pine tree. I think that's a longleaf pine. All right, now back to spore production for a second. I got some terminology we got to go over so that when we get into the future and talk about this, we won't get confused. So across the top, homosporous spore production. Homosporous spores occur in most seedless plants. Actually, this was the moss in the reproduction. Remember the moss? I said the spore is going to produce a gametophyte. So in the seedless plant, we have a sporangium and a sporophyll producing one type of spore, and this is a bisexual spore. What we mean by a bisexual spore is a bisexual spore produces a bisexual gametophyte. This was the thing we were looking at in the fern. That's what I was trying to get at just a second ago, and I drew a vapor lock. The fern, that heart-shaped prothallus, that was a bisexual gametophyte. Because in a bisexual gametophyte, you both have the male and the female on the same gametophyte. Mosses are that way too. On those little green wispy things that stick up, on that same gametophyte, there's going to be male and female parts because they come from one type of spore that gives rise to a gametophyte with both types of sex organs on it, if you will. Sperm and egg, Antheridium and Archegonium. In heterospores, you make two different kinds of spores. You make a bigger spore, which is the mega, the mega that you see there, and you make a smaller spore. Now, this happens in seed plants. So this happens not in the plants we've talked about so far, but in the pine trees and in the flower and the fruiting plants. So let's go through the terminology here first. Megasporangium are megasporophylls. Megasporophyll is a modified leaf. In the case of a pine tree, a megasporophyll are the female pine cones. These are the female pine cones, the bigger ones here. So that female pine cone is a megasporophyll. On that megasporophyll, we'll see this in lab when we get back, are megasporangium. And megasporangium produce megaspores. And megaspores produce female gametophytes. Now, this is no different than anything in alternation of generation. You get a sporangium, it produces spores, and spores that produce a gametophyte. A gametophyte that produces the egg in this case. In the case of the males, everything is smaller. That's the micro. Microsporangium, or male cones in the case of this pine tree. This is the male cone. These are male cones. Right, there's two different types of pine cones on a tree. Have microsporangium made on microsporophyll. So the cone is the microsporophyll, make microspores, they make the male gametophyte. And of course, as I said, that produces the sperm. Okay? Now, we're going to see these terms a lot, especially when we talk about them in lab, so make sure you take a look at them and you're familiar. Just remember, mega is going to be female. Like the female egg, even in animals, is almost always, I believe, bigger than the sperm in all cases. I'm not aware of where it's the other way around. All right, so now let's go into the vascular plants. All right, so now we're into the tracheids. And first group of tracheids we're going to deal with are the tracheids that have xylem, and they have phloem, but now they are still seedless, right? They're still seedless. We don't have flowers and fruits and seeds until we get the last two group of plants, which is the subject of the next chapter. So we're going to look at the lycophytes first. So the part that's right there in the red, the lycophytes, the club mosses, the spike mosses, and the quillworts. Now I put moss in quotes to remind you again that these are not true mosses. These are not bryophytes. These are not non-vascular. Some of these are epiphytes. Epiphyte plants live on other plants. Orchids are epiphytes, for example. Spanish moss is another epiphyte. Mosses, again, because they look that way. It's all about this idea of convergent evolution, right? It was these organisms that were real prominent back during the Carboniferous. You see in the picture on the right, I always love going out online trying to find pictures of what they think it looked like. This is what it looked like about 300 million years ago or so during the Carboniferous period. It's called the Carboniferous because of the fact that there was so much plant growth going on during this time that there was a lot of carbon, right? A lot of organic material. Matter of fact, this is where where a lot of the coal comes from. So the lycophytes, there's some ferns and some horsetails. There's some lycophytes right there. Lycophytes were really big. In some cases, they were tree-like. Now they're much smaller. They're much smaller now. This is since the Carboniferous period. But the club mosses are homosporous, right? So they're going to make a bisexual gametophyte, just like the ferns do. The spike mosses, which you see there, and the quillworts are heterosporous. So they're going to have a megaspore and a microspore. The manilophytes. Oh, in your book or in your lab, in your field notebooks, it calls the manilophytes are the pterophytes. So pterophytes and manilophytes are the same. The ferns, the horsetails, and the whisk ferns. All right. Now, interestingly enough, the manilophytes are not as closely related to the lycophytes as they are to the seeded plants. So this connection right here, these guys are actually more of a sister group to the flowers and fruits and the cone plants that we'll talk about next time, even though they're considered in this seedless vascular group of plants. So it's the ferns, the horsetails that we looked at in the lab, and there's a whisk fern. They don't get very big. Like I said, if you imagine back to that discussion about leaves, some whisk fern that evolved through time and all those branches were in the same plane, and then you put the material between it, you could make leaves. So who do we want to look at first? Is it the ferns that we look at? Yeah, ferns. All right, the ferns are a very widespread group, about 12,000 species. Most of these guys being homosporous, a bisexual gametophyte. Now, again, make the connection. Remember, that's what we looked at in lab. We looked at a fern gametophyte, that little heart-shaped thing. The male and female parts were on the same gametophyte, so that means it's homosporous. Trillions and trillions of spores. Now, a lot of times ferns have the sori on the back of the frond, like I showed you, but there are some species that don't, like the one in the upper right. This is a Florida species. This is known as a cinnamon fern. The cinnamon name comes from the fact that the spores aren't produced on the back of the fern fronds. They're actually produced on a separate specialized frond, this thing right here. These are the spores, and since they take on that kind of brown color, they kind of look like cinnamon, so that's where they get their name, cinnamon ferns. Ferns are known for a very conspicuous, very large, like in that case of the picture there in the background, the Australian tree ferns, very big sporophyte, very small gametophytes. Very small gametophytes. All right, now, alternation of generation in ferns. Let's start with what we know. Let's go down here to the bisexual gametophyte. All right. The bisexual gametophyte has got Antheridia and Archegonium, so it's got both of those, gamete-producing structures on the same structure. That's why it's a bisexual gametophyte. The Antheridia produces flagellated sperm, which swim through water. So ferns are one of those that produce the egg and the sperm at different times to prevent inbreeding. Sperm finds an egg nonetheless and forms a zygote, and out of the zygote, now this is all happening on the underside. When you were looking at that under the microscope, you were looking at the gametophyte upside down, because all this is happening on the underside. See these little rhizoids? That word rootlet really shouldn't be there. So coming out of that archegonium is the zygote, which turns the corner and grows up between that little area of the heart shape on the gametophyte, and that's our new sporophyte, and that's going to become this. And they're really kind of cool. They unroll. So first they unroll longwise, and then the leaves roll out from the center stem. They call it a fiddlehead, a young leaf, because it looks like the part of a violin that you tune, a fiddlehead. And then on the other side of those, there are the sorus, right? If we're talking about one, and inside of those, there are sporangium, and sporangium are producing spores through meiosis. So once again, you see that word meiosis, and you should immediately think, oh, we're producing different types of spores, and then through mitosis, we're going to produce lots of them. There's the horsetail again with the elater. Right, and the spores will disperse, and if they find a place to settle down, they will grow back up into, and there's only one kind of spore. These are homospores, so there's only one type of spore. And the process is repeated, right? What's this look like in real life? I found some pictures and put together. So here's the Antheridia. There's the sperm. Here's looking down the archegonia, looking down the vase, and there's an egg at the bottom. Out of that would grow the new sporophyte, which would first start off as a fiddlehead. It would continue to unroll, and then the leaves would unroll, and on the back of those are the sori, which are made of the sporangium, which produce lots of spores, which in turn produce the gametophyte. I have all this labeled too in the slides. I think we're going to finish chapter 29 today. I was trying to look at my, let's see, do 29. We'll do 30. We can do 30 tomorrow. We can do 30 next week. Oh, let me think about that for a second. All right. All right, so now we got the horsetails left in this group. Very diverse during the Carboniferous. Now there's only about 15 species in one genus, Equisetum. Horsetails, remember Equus? I showed you yesterday they have the vegetative stems, little tiny leaves. Those are examples of lycophiles, little tiny leaves. And then the other stem, kind of like in the cinnamon fern, there's a fertile stem on there as well. All horsetails are homosporous, so we're going to produce a bisexual gametophyte, and these were the ones you remember had the silica. They're called scouring rushes. Rush is a type of plant, and these are called scouring rushes because of the roughness of the silica. Whisk ferns, again, very small, subtropical. Living fossils. Now, living fossils, again, are things that we thought were dead but are still alive. Like sometimes they discover, oh, we thought this plant had gone extinct, millions of years ago, and you find it on some remote mountaintop in, South America, something like that. Or organisms that are not ancient organisms, but look like ancient organisms. Whisk ferns kind of look like some of the early vascular plants. That's why we call them living fossils. But they're actually more closely related to ferns than they are to those early vascular plants. No true roots. They're subtropical and tropical plants. So oftentimes in the subtropics, organisms get most of their nutrition from the photosynthesis. There's not a lot in the soil in the tropics because of the decomposition rate is so fast. Things decompose so quick in the tropics that things get sucked up into the living plant matter very quickly. So these guys basically have no true roots. All right, so where are we? As we move into the end of the Carboniferous during this time of extensive forestation, the carbon dioxide levels begin to drop, right? More trees, more organisms photosynthesizing, less CO2. So carbon dioxide levels begin to drop. And when carbon dioxide levels begin to drop, we get the opposite of what we see in global climate change. Right, we talk about carbon dioxide levels rising in the atmosphere, trapping heat, causing the temperatures to rise on Earth. If the opposite happens, right, and I know some folks think out there, well, isn't that what will happen with global climate change? We're putting much more carbon dioxide into the atmosphere than any amount of plant growth could deal with. But back during this time in the Carboniferous, before humans were monkeying with all this, carbon dioxide levels begin to drop during the Carboniferous because of all the plants. So global temperatures begin to drop as well, begin to get cooler. In addition to that, there was a massive diversification of marine organisms, especially those that have things like shells, like you see in the picture down below, and they need a lot of calcium carbonate. Remember I talked about calcium carbonate? You find it in the exoskeleton of corals, and you find it in the shells of scallops and oysters, and that's a very common structural element in the ocean, this calcium carbonate. So a lot of the atmospheric carbon was being removed into the ocean. These ocean organisms were sucking up this atmospheric carbon to make this calcium carbonate in order to produce all these shells. So we started getting this cooling off, and so the plants begin to die. Lots of plants begin to die, and we got so many. We get this. That's why it's called the Carboniferous. If you dig down in the rock layer, when you get to the Carboniferous, 300, 400 million years ago, you get this really thick layer of organic material, coal. That's what coal is. See in the picture on the right, peat, which is these plants die and they form that peat moss. Through heat and pressure becomes lignite, which eventually, through more heat and pressure and time, obviously becomes coal. That's where, like in Pennsylvania, where the big coal mines are, that's a huge Carboniferous place. That's where there were a lot of trees during the Carboniferous. 6 million tons a year of coal we burn, dumping carbon dioxide in the atmosphere. Coal, there's no such thing as clean coal, by the way. So, all right, toward the end of the Carboniferous, the Earth begins to dry up. All right, and when you dry up, things that allow you to survive this drying up are going to be favored. So seeds would have been favored. Seeds are actually better at surviving than spores, so there would have been a more of an emphasis on seed plant evolution, and that's, of course, where we'll move into next. If you see down below in that little chart, the temperature and the carbon dioxide levels dropping at the end of the Carboniferous. Soon, the carbon dioxide levels are going to begin to rise, the temperature is going to get warmer, and the environment's going to get drier. And all of that, is with the die-off in the Carboniferous and the climate getting drier, is going to, again, favor those seed plants of dominance. But among the animals, and I know we're not talking about animals here, we're getting ready to move into the era which the dinosaurs dominated. So when the environments begin to dry up, you get those, think about it in the plants. You got the seed plants when the earth is drying up, figuring out how to survive by producing durable seeds, taking care of their young, right? And you also got the animals doing that with the reptiles, having the eggs. They don't have to lay in water anymore. It's remarkable, right? It's all about the environment that drives all this. I've talked about this since the beginning of the semester, haven't I? It's all about the environment that drives organism evolution. Doesn't matter if you're a plant or you're a dinosaur, you're still going to have to deal with the same concerns. All right, does anybody have any questions for me? I want to look at the schedule for a second because, let's see, today's the 24th. You guys have Memorial Day. Next, I have 29. I just did 29. All I got to do is 30. All right, we're not going to have lecture tomorrow. I'll post a notice on Blackboard. I'll give you another day to study for the lab practical. I will be online tomorrow during lecture time between 1 and 2:30 for sort of like an office hours if you have any questions. But we will not hold lecture tomorrow. There will not be lecture. We'll do chapter 30 next week after we get back from Memorial Day. So study for the lab practical and let me know if you have any questions, okay? All right. Let me stop the recording.